Hi guys, in animal kingdom we study about different animals found on this earth. If you see all the animals, then out of them around 70% of the animals are actually the insects. So in this chapter we'll be focusing on the characteristics of different types of animals like the lizards, the humans, and we can say the fish, frogs, means different kinds of animals we'll be seeing. Their general characters will be starting in this chapter. So the kingdom Animalia, which is the fifth kingdom of Whitaker's classification, it actually comprises of all the multicellular heterotrophic organisms which do not produce their own food and they do not have cell wall present in them. Clear? But we have one more member to study here, that is known as the primitive members of the Animalia. We can say they are the ancestors of kingdom Animalia and the group is Protozoans. So guys, if you remember in chapter 2, that is in the biological classification, we discussed the second kingdom, that is kingdom Protista, and in that kingdom I told you that we'll be studying this particular group of Protista in the kingdom Animalia. So you won't have to get confused that Protozoa are included in which kingdom. It's very clean and clear that Protozoans are included in the kingdom Protista, but we are studying it here because they have quite similar characteristics to the kingdom Animalia. Clear? Now, regarding this chapter, Kingdom Animalia, sometimes students do get confused because of lots of characteristics and lots of examples. They think like if we talk about the mammals only, then there are a number of examples in our general life also, but you won't have to remember all the scientific names and all the examples in this chapter. Some standard examples are there, only those standard examples actually you guys have to learn, and the questions are always asked from those standard examples only. So you won't have to get worried about the examples. I'll make it sure that once you complete watching this video, we can say all the videos of this chapter, you can score complete marks, full marks from the questions asked from this chapter. So first we'll talk about the group Protozoa that are from the kingdom Protista. I'm again and again repeating so that you guys won't get confused. And then we'll see how the classification of kingdom Animalia is done. So in this video, we'll be focusing on two parts. First is Protozoa and second is basis of classification of animals. So now let's get started. So guys, these protozoans, all the members are actually heterotrophs, means they will not produce their own food and they will depend on other organisms for their food. So let's write first point, that is all are heterotrophs. Clear? Now coming to the next point, the protozoans can be parasitic also or they can be predators also. It means that either they will hunt the organism, means they will hunt their prey and feed on it to get the nutrition, or the possibility is that some of the protozoans will live on some other organism that is known as host, will not kill that host but they will just get the nutrients and keep harming the host. So protozoans can be parasitic also or they can be predators also. So we have the next point with us, parasites or parasitic or predators. Clear? Now, next point, these are known as primitive relatives of the animals because they are having many similar characteristics to the animals. These are known as primitive relatives of animals. Primitive relative of animals. But they belong to which kingdom? They belong to kingdom Protista. Clear? So you won't have to get confused with this point, guys. Now, the four major groups are there into which the protozoans are actually classified. That is ciliated, flagellated, then we have amoeboid, and the last one is spore forming. So the first we have got is amoeboid, number one. Amoeboid, these organisms will have some amoeba-like structure, means the common example is their amoeba. These organisms or protozoans can be found in fresh water, sea water, or in the moist soil also. So what are the habitats of the amoeboid protozoans? Fresh water, or they can be found in marine water or sea water, we can say, to be more exact, sea water, or in the moist soil. So these can be found in these different habitats, capture their prey to move from one place to another. They have some specific structures which are known as pseudopodia. So amoeboid protozoans have a specific structure which is known as pseudopodia, which helps to capture the prey. In your 9th standard, you might have studied that nutrition in amoeba. So guys, for nutrition in amoeba, those amoeba cells will just move towards the nutrition, will trap the food, will move towards the food, will trap the food. So to move, they use some finger-like projections which are known as pseudopodia. These pseudopodia are actually known as false feet. They are known as false feet. Clear? Now, these marine forms, means those amoeboid protozoans which are found in the marine habitat, they have silica depositions or silica shells on their surface. So next point, marine members will have what? They will have silica shell. On the surface. Two examples are there. One is ant amoeba and another one is amoeba. And ant amoeba is a parasite, means it will live on the host organism and will harm the host. So examples, amoeba, and another one we have is ant amoeba. Clear? Now, after this we have the second member of the protozoa, that is flagellated protozoans. So number 2 is flagellated. Flagellated means flagella will be there in these protozoans, and the members are either free living or parasites. Either they'll be not depending on other organisms, or they can be the parasites also. So the first point we have got is free living or parasites. Free living or parasites. Direct questions are formed in the exams. So you won't have to go into the much details of these points. You just need to remember these points. Parasites. Clear? Now, these organisms have flagella, and the parasitic forms causes diseases, and the most common example is in number of times in a number of exams, that is trypanosoma. Example is trypanosoma. Trypanosoma causes sleeping sickness. This is the most common question from this particular topic, that is protozoa. Who causes sleeping sickness? Disease caused by trypanosoma. So this point is very, very important. Sleeping sickness, I'll give it 2 star because it is very important, trypanosoma causing sleeping sickness. Now, the vector which transmit this protozoa and to cause the disease is known as CC fly. It is CSE, GSE, CC fly. So you won't have questions from this fly name, but you will get the name from this protozoa and the disease name specifically. Then we have the third member, that is the ciliated protozoans. Ciliated protozoans means this time what will be present on the surface of the cell? Cilia will be present, small hair-like structures will be present. A group of cilia beat in a coordinated form, and they help for getting the nutrition, they help for the locomotion. Now these members means the ciliated protozoans have a cavity which is known as gullet. So they have cavity, and this cavity is given a name gullet. Now this gullet is actually used to store the food. The cilia will send the food and the food will get stored within the gullet. Clear? So what is the function of gullet? To store food. Store food. Right guys? Now example is there with us, that is paramecium. Example is paramecium. And some of the members of this protozoa, for example, paramecium, show nuclear dimorphism. Nuclear dimorphism means more than one type of nucleus will be there. For example, in paramecium, two types of nucleus are there. One is macronucleus and another one is micronucleus. So such phenomenon where different types of nucleus are there, it is known as nuclear polymorphism. Here specifically in paramecium, two types of nucleus are there. So nucleus dimorphism is there. Now next is number 4, that is sporozoans. Sporozoans. Sporozoans are those protozoans which have spore formation in their life cycle. So all the spore forming stages are means spore-like stages will be there in the life cycle of the protozoans. Those are placed in this group. The most common one and the most notorious protozoa, yes, the question is asked from this point, most notorious. The most notorious member is it is Plasmodium. Plasmodium. Now this Plasmodium causes malaria disease. Different species of Plasmodium are there, like Plasmodium falciparum. It causes malaria. So guys, we say malaria causing mosquitoes. Actually, those mosquitoes just carry these Plasmodium. Mosquitoes are not the cause, but they are just a carriers. They just transfer Plasmodium into our body, which causes malaria. So who is the vector over here? The vector is female Anopheles. Female Anopheles is what? Female Anopheles is actually a mosquito. This female Anopheles will transmit the Plasmodium into our body to cause malaria. Male Anopheles are not responsible for the transfer of malaria. So guys, keep yourself safe from the female Anopheles specifically. In detail about this Plasmodium, malaria, female Anopheles and the complete life cycle of Plasmodium, you'll be studying in your 12th standard, that is disease chapter. So we'll just end up here with the example. Here we are done with the group Protozoa, which is belonging to the kingdom Protista. You must be thinking I have been repeating the same point again and again because I don't want you to get confused. Plus, I want you to learn everything here in the video itself so that next time when you are going through the book or any other material, you will just be revising because revisions are very important. So here we are done with this first part of what we are going to do today in this video. Next, we are going to see how the plant kingdom, how the animal kingdom is actually classified. So guys, we have the different criteria which are used for the classification of kingdom Animalia. First, we'll understand all those criteria. Then I'll tell you the line diagram through which you can remember the complete format, the complete classification of different phylums. So we'll make it very easy, very simple to remember. But first we need to understand so that we can easily remember also. So the first one we have got is levels of organization. Number one is levels of organization. Levels of organization. So guys, you know that in kingdom Animalia, all the organisms are multicellular. Means a single organism will be made up of more than one cell. But it is possible that those group of cells are not forming the tissue or they are forming the tissue. So on the basis of levels of organization, we have two partitions. One is cellular level where the organism will be made up of more than one cell, but those cells will not form the tissue. Those cells will together present, but they are not forming the tissue. This is known as cellular level. We'll see the examples also. That means the phyla which have the cellular level when we'll see the line diagram of this complete classification. Next group is tissue level, organ level or organ system level. Tissue level, organ level or organ system. Means all those organisms or the animals which have tissue formation or the tissues together forming the organs or organs together forming the organ system. So this level of organization is found in different phyla which we'll be seeing in the line diagram. After this we have the second basis of classification, that is number 2, and it is symmetry. Symmetry we study in maths in our junior classes. Now we are studying symmetry in this animal diversity. Further we'll be studying symmetry in the plant morphology also guys. But the basics are same for the symmetry. So let's put the heading symmetry. Symmetry means when you divide an animal, it should divide into equal half pieces. Now, on the basis of symmetry, we have two types. One is radial. One is radial and another one is bilateral. Radial means you can cut it with any axis passing through the center. It will divide into two equal half pieces. For example, you can take a cylindrical structure, or we can say the cylindrates, and you can divide this into half pieces with any plane, right? And this type of symmetry is known as the radial symmetry. Whereas coming to the bilateral symmetry, the organism can be divided into equal half pieces only with a single plane. With one lateral line, we can divide an organism into two half pieces. So this is the bilateral type of symmetry. Next we have is germ layers. So guys, on the basis of germ layers, the organisms can be the diploblastic or they can be triploblastic. So let's put the heading first, germ layers. Germ layers means ectoderm, mesoderm and endoderm. So on the basis of presence of these three layers in the organism, two groups are there. One is diploblastic, and the another one we have is triploblastic. Triploblastic. Now, what is this diploblastic and triploblastic? So, in case of diploblastic, three layers are there. That is the ectoderm, plus another one we have is endoderm, and in between this ectoderm and endoderm, undifferentiated cells are present which are known as mesoglia. So guys, two layers or two dermal layers are there surrounding an undifferentiated layer of cells or tissue, and that we call as mesoglia. Whereas in case of triploblastic, the three derm layers or germinal layers are present: ectoderm, next is mesoderm, and the last one is endoderm. So guys, the lower animals have diploblastic, or the higher organisms are triploblastic. It means they will have three germinal layers, those which are triploblastic. Some of you might be wondering, what is this ectoderm? What is this endoderm? So guys, ectoderm is the outer covering or present on the outer surface. This is the germ layer which is forming the outer surface of the body, whereas endoderm is forming the inner surface of the body. Clear? Now after germ layers, we have the fourth point, that is body plan. Number 4, body plan. So guys, different body plans are found in different types of animals. For example, in some of the members of this kingdom, they just have cells aggregation. So cell aggregates means the cell will be present together in the form of aggregation without formation of any particular type of structure, without any tissue formation. Then there is blind sac. Blind sac, you can say that there is just one opening. Simply we can say a tube with one opening. For example, test tube. A test tube will have only one opening. Such type of alimentary canal which has only one opening. An alimentary canal which has only one opening, that is known as blind sac. All these terminologies you will be getting in the different phyla we are going to study in this chapter in the upcoming videos. So guys, what is blind sac? Let's say again, blind sac is an alimentary canal which has only one opening. Clear? Next is tube within tube. Tube within tube. What does it mean? It means that the digestive system will be open from both the sides. Just like we have mouth and we have anus as opening, and means the opening means starting and ending of the digestive system, to be more exact. So tube within tube means the digestive system will be there having opening on both the sides. So in us we have mouth and the anus. So guys, this is the tube within tube we have as the body plan. Now in case of tube within tube also two types are there. You will be getting these terms First one is protostome. Protostome, it means that in the development, means during the embryo development, the mouth develops before the anus. Whereas another term you will get is deuterostome. So in the complete an alimentary canal, the anus development takes first, then the mouth develops. This is known as deuterostome. Clear? So when anus develops before mouth, we call it as deuterostomes, and when the mouth develops before anus, we call it as protostome. So lower animals are actually the protostomes and the higher ones like we are the deuterostomes. Next we have is body cavity or coelom. So number 5 is body cavity. Terminologies are definitely important, guys. Coelom. But you won't have to learn anything for these terminologies. Just get the things clear in your mind and that's enough for you. So between the intestinal canal and body wall, there is present some cavity which is known as coelom. So between our digestive system and the body wall, there is means the intestinal canal and the body wall, there is some coelom, some space is present, some cavity is present that is known as body cavity, also known as coelom. The coelom is actually a fluid-filled cavity present between intestinal canal and body wall. So sometimes we just say coelom absent, coelom present, coelomates, pseudosalomones, acoelomates, but we don't know what is coelom, right? So first we need to know what is coelom, what we are talking about. So what is coelom? Coelom is a fluid-filled fluid-filled cavity between the intestinal canal, means the alimentary canal we can say, and body wall. So if I take the transverse section of my body, let's say this is the intestinal canal and then we have this body wall here, right? So in between that some cavity is present, you can see that cavity is actually the coelom. So I'll erase this part, is actually not required. Okay. So next point is that they function to protect the internal organs. For example, our heart. Our heart has its separate covering, pericardium, right? Then lungs have their own coverings. So to protect these internal organs, they have the coelom, means protective cushion. So protect internal internal organs. Then they are present as skeleton in case of worm. So three forms of body cavity or coeloms are there. Number 1 is acoelomate. If a word is present, what does it mean? It means that absence. So in case of acoelomates, no body cavity is present. No cavity is present between the body wall and the digestive tract. So how does that transverse section would look like? I'll show you here on the screen. So let's write the first point. What is this acoelomates? No cavity. And how will that transverse section look like? It would look like somewhat like this. So this is the gut. Then we have the endoderm, which is lining the gut. Then after endoderm, we have the second germ layer. Remember, acylomate, pseudosalomates will talk in the case of triploblastic, not in the case of diploblastic organisms, because mesoglea is itself a undifferentiated layer of cells, right? So this red colored part is what? Red colored part is mesoderm. Then I'll draw the green colored part, that is the ectoderm. So you can see there is no body cavity present around or in between the gut and the body wall. So this is ectoderm. Then we have the red colored part, that is mesoderm. If you remember these layers now, you won't have to learn anything, right? Then inner one is endoderm. Just keep this diagram in the mind, that's all you need to do. And this is gut, means the intestinal canal or alimentary canal, digestive tract, anything you can call it. So there is no cavity present in this particular type of organisms. Clear? So these are the acylomate types. Next we have is B, pseudosalomates. Pseudo means what? Actually they are not, but they look like it, right? So next is pseudosalomates. Pseudosalomates will have the cavity partially lined by the mesoderm. So I'll show you how they look like, but let's write what the pseudosalomates are. Cavity partially lined by mesoderm. Means it will not be lined on both the sides by the mesoderm. Partially lined by mesoderm. Clear? So how it would look like, I'll show you now on the screen. This is again the gut, and the gut is surrounded by endoderm, right? Now, after this endoderm, we have some cavity present, and on the outer side, there will be these mesodermal cells present like this. So this red colored structure is actually the mesoderm, and outer to that is the ectoderm. This is ectoderm. Then the red colored part is mesoderm. I'll draw the diagrams 3 times, so you will be learning all the germ layers definitely here itself. Then this white space you can see is actually the body cavity. It is body cavity, or we can also call it a coelom. Then this black layer is the endoderm, and it is surrounding the gut. So you can see that only on the outer side the mesoderm is present. Mesoderm is not present on the inner side. There is no layer present here of the mesoderm. Hence these organisms are known as pseudosalomates, because they will have partial lining of the mesoderm. Then we have C, or number 3 type of organisms, which are known as Usalomates. Usalomates means they are true coelomates. They will have true body cavity in them. So true body cavity means what? They will have the mesoderm lined on both the sides. So first point regarding usalomates is true coelom, and they have the mesoderm which provides cellular lining, which is known as peritoneum. So mesoderm forms a lining which is known as peritoneum. Clear? So how the organism would look like, let's draw it. So this is the gut surrounded by the endoderm. Then on the inner side, what is present? Mesoderm is present. This is what? This is mesoderm. Then again on the outer side, what will be present? Mesoderm will be present. This is mesoderm, and the mesoderm will be surrounded by the ectoderm. Right? So you can see that this cavity is actually lined by the mesoderm on both the sides, hence it is known as true cavity. Now the cavity formation is further of two types and hence the classification done. First one is schizosylom. Again the terms are important actually. So first we have got is schizosylom. Here, the cavity is formed by the splitting of mesoderm. Mesoderm layers are there like this and they will split to form the cavity. When the mesoderm split to form the cavity, the cavity formed will be known as schizosylom. So let's write what does it mean. Cavity formed by splitting of mesoderm. Splitting of mesoderm. Splitting means separation of mesoderm. Half of the layers will means this is the complete mesoderm, half will go this side and half will go this side, forming a cavity in between them. Then we have is enterocylom. Enterocylom means cavity is formed by the fusion of patches formed in between the mesoderm during embryonic development. So initially, few patches will be formed. We can say the form of patches will be there. Those patches will fuse to form a complete body cavity that will be known as enterocylom. So these patches are formed during the embryonic development which later fuse to form the complete coelom or true coelom. So this is all about the coelom or body cavity. Next we have got is number 6, segmentation. So guys, body is externally and internally divided into segments. These segments are also known as metameres without any repetition of some of the organs. Clear? So in case of the segmented organisms, the segments will be there, or we call the segments as metameres also. Number 7 is notochord. Notochord is a mesodermally derived part. So first point is mesodermally derived. Means it is derived or it is formed by mesoderm. It is a rod-like structure which forms the mid-dorsal surface during embryonic development. So on the basis of presence or absence of notochord, we have the non-chordates and chordates. Non-chordates do not have notochord, chordates have notochords. So what are these notochords? These are rod-like structures. These questions are actually asked in the exams. Rod-like structure where they are formed on the mid-dorsal surface. Mid-dorsal. In detail about this notochord, we'll see when we'll start these chordate members, because non-chordates do not have notochord. So what two types are there? Let's write those names also. One is non-chordates, the lower animals, and another one is chordates. Right? Now coming to the next point, that is blood vascular system. So guys, not all animals have blood actually. There are some organisms which do not have blood. There are some organisms which do not have blood vessels like us. So those who do not have blood vessels like us are said to have open blood circular system, whereas we who have proper vessels, proper tubules in which the blood flow, we are known to have closed blood vascular system. So number 8 is blood vascular system. And what are the two groups? One is open, where no proper tubules are there, just all the organs are placed within, ba- like placed within the blood itself. And next is closed type, where proper vessels are there and blood will flow only through those vessels, not out of those vessels. So guys, these are actually the basis of classification we have for this animal kingdom. Now we'll see the line diagram. We'll be revising all the points, seeing how the different phyla are actually placed. So let's write first point, kingdom. Animalia. This kingdom Animalia is divided into two groups on the basis of levels of organization. On the basis of what? On the basis of levels of organization. What were the two levels we saw? The two levels we saw, first one was cellular and another one was tissue level or organ level or organ system level. The organ systems will form the complete organism, right? So this cellular ones have the example, means the first phyla, that is phylum Porifera. So the Porifera members, which we'll be seeing in the next video, they do not have the tissue formation, a number of cells present together forming the body, but not the tissues. Coming to this tissue organ system level of organization, they are further classified on the basis of symmetry. On the basis of symmetry, again we have got the two groups. First one is radial symmetry. If you learn this diagram, these points can be easily recalled in the exams. You won't have to do extra work for this classification. And another one is bilateral. Clear? Now, the radials have two phylum present in them. Number 2 phylum is Coelenterate. The coelenterates are also known as Cnidaria. Clear? So don't get confused with the names. Number 3 phylum, or the phylum, second phylum which is included in the radial symmetry is Ctenophora. This is a small phylum. Rest all the phylums we'll see are the major phyla. Ctenophora is a small phylum. Coming to the bilateral symmetry, let's see the further classification. So here in the case of bilateral classification or bilateral symmetry, now we'll talk about the coelom. Means on the basis of coelom. So guys, did you notice one point? I didn't tell about diploblastic and triploblastic. Reason. We talk about coelom only in the case of triploblastic organisms. We do not talk about coelom in the diploblastic organisms. Clear? So triploblastic organisms starts here with the bilateral ones, and on the basis of coelom or body cavity, we have the three groups, that is acoelomates, Then we have pseudosalomates, and the last one we have got is salomates or Usalomones. So let's see different phyla which are placed on these different Coelomate types or Coelom types. So Acoelomates includes Platyhelminthes, number 4, Platyhelminthes. Clear? Next is Pseudosalomates, which do not have true coelom, or we can call them to have false coelom. Number 5 phylum is Aschelminthes. Then starting with Annelida means number 6. Annelida, then we have Arthropoda, then we have Mollusca, then we have Echinodermata, then we have Hemichordata, and the last one we have is Chordata. So let's give the numberings also: number 7, number 8, number 9, 10, and 11. So guys, these are the major phyla, including Ctenophora, which is a minor phyla, and this is the complete line diagram which you need to learn. I'll also write one term here so that you will remember about the diploblastic and triploblastic. That is coelomates we talk means we're talking about the triploblastic animals only. Triploblastic. Clear? So this way you can easily remember what the basis of classification is. In the upcoming videos, we'll be discussing about these phyla separately. We'll be talking about much more characters about these phyla which are asked in the exams. So guys, stay tuned, and for that you will have to subscribe my channel. Please like this video if you liked it actually, and comment below what else do you want to know regarding the animals apart from this exams point of view. I'll definitely get it for you, and please share it with your friends too.